Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Syahadah binti Abdul Malik from Cas12a. Today I would like to present a CRISPR Cas12a-assisted array for Helicobacter pylori DNA analysis in saliva. Helicobacter pylori is a bacterium that infects the stomach and is a major cause of peptic ulcer and gastritis. It also linked to an increase of risk stomach cancer and lymphoma. H. pylori infections is common with the majority of the infected individual being asymptomatic. In some cases, it can lead to the serious complications. However, the development of CRISPR's Cas12a-assisted array for the analysis of H. pylori DNA in saliva samples is significant as it addresses the urgent need for an efficient and convenient approach to accurately diagnose H. pylori infections. The use of saliva samples for the detections of H. pylori is promising as it offers a non-invasive and easily accessible method of diagnosis. The research on the CRISPR's Cas12a-assisted array for H. pylori DNA analysis in saliva samples demonstrate the potentials for rapid and sensitive detections of the bacterium, which could lead to the development of the non-invasive and diagnostic tools for H. pylori infection. The objective of this study is to aim to develop a CRISPR Cas12a-assisted array for the analysis of the saliva sample to detect and diagnose H. pylori infections. And second, the array is designed to have integrated H. pylori concentration detections and genotype screening function. It allows for the accurate detections and classifications of the different types of H. pylori genotype. And the third is the study is to aim to address the challenges associated with saliva sampled analysis by employing a strand displacement amplifications (SDA) strategy to improve the sensitivity and the accuracy of H. pylori DNA detections in saliva sample. The method that being used in this study is first, CRISPR Cas12a system constructions and optimizations. These constructions of prop for CRISPR-12 The methodology of this study is first, CRISPR-Cas12a system constructions and optimizations. It is to construct a prop for CRISPR-12 Cas12 trans-cleavage reactions, and it's to identifications of the different nucleic acid chains by the constructed props. Second, screening array development. The development of a screening array for high-throughput detections of the different types of H. pylori genotype using different props. Third is this SDA integrated CRISPR's Cas12a systems. It's the integrations of the strand displacement amplification SDA strategy with CRISPR Cas12a system to improve the sensitivity and ensure the accurate diagnosis. The fourth method is detections of actual saliva samples. It is the evaluations of the sampling quality using housekeeping genes, which is the housekeeping genes that being used is glyceroldehyde 3-phosphate dehydrogenase, which is GOFT. It is to validations of the approach using synthetic sequences of H. pylori in PCR experiment and establishments of standard curve for quantitative analysis. The fifth method is analysis and validations, which is the principal components analysis PCA for analyzing the response patterns of the screening array. And the sixth method is statistical analysis, which is the analysis of the fluorescence signals and the PCR result for validations of the approach. Result and discussions. For this figure, it shows the principles of the CRISPR's Cas12a system. The figure illustrates the basic principles of the CRISPR's Cas12a system, which is it involves the trans-cleavage of the specific label nucleic acid reporter in the represents of the detectable target sequence. Next is the optimizations of the CRISPR-Cas12a. The figure demonstrates the impact of various factors such as temperature, nucleus concentration, reactions time, and concentrations of reporter DNA and cRNA on the efficiencies and performance of CRISPR's Cas12a system. Next, a screening process of diverse genotype. From this figure, it illustrates the process of high-throughput detections of different genotypes by the array, demonstrating the system's ability to distinguish between different genotypes, then probe recognitions and principal component analysis. The figure shows the recognitions of different nucleic acid strands by the constructs of the probe, and the results of the principal component analysis indicate the system's specificity and the classifications of the accuracy. Then, the strand displacement amplification (SDA) and CRISPR's Cas12a integrations. From the figure, it demonstrates the successful integrations of SDA with the CRISPR-Cas12a system. It shows that the implications of the targeted DNA and the identifications of different genotypes using the SDA-assisted CRISPR-Cas12a system. Then the verifications of the SDA and absorption spectra. The figure shows it provides the evidence of the successful SDA process, as well as the absorption of the spectra of the various components involved in the SDA process. Then, the stability of saliva by PCR. From this figure, it illustrates the stability of the saliva by PCR. It's providing the insight into the quality of the saliva samples and the reliability of the test. Overall, the results from the figures collectively demonstrate the constructions, optimizations, and performance of CRISPR's Cas12a-assisted array for the detections of Helicobacter pylori DNA in saliva. The figures provide evidence of the system specificity, sensitivity, stability, and classifications, accuracy, as well as successful integrations with strands displacement amplifications for accurate diagnosis. In conclusion, the study successfully developed a CRISPR-Cas12a-assisted array for accurate diagnosis of Helicobacter pylori H. pylori infections in saliva sampled. The integration of H. pylori concentration detections and genotype screening functions within the array allowed for differentiations of 10 different H. pylori genotypes, demonstrating its potential for comprehensive analysis. The combinations of an isotomer cycling strategy with trans-cleavage activity of Cas12a and implementations of trans-displacement amplification SDA contribute to the assay high sensitivity with a low limit of detections LOD of 60 FN. The validations of the array's performance using clinical saliva samples from H. pylori infected patients further confirms its effectiveness. It shows the good sensitivity, the selectivity, and the stability, as well as the reproducibility. That's all for me. Thank you for watching.